Share screen. Oh, nope, that's the scene. Okay.
Nice.
All right, Nikki, you are our sole viewer right now, but for anyone else watching, welcome to Protein Trafficking and Intro to Gene Expression. Both Sebastian and I are going to go through it. Yeah. So we'll start with protein trafficking. So an important part of protein trafficking is that proteins start translating on a free ribosome in the cytoplasm. So that all the proteins that you have originally will end up in will be made in the cytoplasm on a ribosome. So every time a protein is localized to the ER, it must have an ER signal sequence. So signal sequences are kind of like, I like to think of them as like a zip code. They tell you where your protein's going to end up. And the signal sequence is kind of it's translated with the protein. So it's like it's a piece of the protein. It's usually on the N terminus, but I think we'll go through that in a few slides. But so basically, if you want your protein to end up in the ER, if that's where its job is, or anywhere in the endomembrane system, it must have this ER zip code or a sequence of amino acids that indicate that it needs to go to the ER. So this is because the ER signal sequence is recognized by the signal recognition particle, which binds to the newly made protein and ships it off to the ER like as soon as it's made. If they have it, the localization sequence, the ribosome will dock to the ER, where it is immediately translocated into the ER lumen. So remember this, just to highlight, this only happens if the protein being made has an ER signal sequence. If it does not have an ER signal sequence, so say its job doesn't have anything to do with the endomembrane system, then it will not leave the cytosol because that's where it's made. It will stay in the cytosol after it has been translated. So basically, if there's no zip code, the protein just knows to stay in the cytosol because that's where it's being made or translated. And then any protein that needs to be sent outside of the cell has to go through the endomembrane system. So it needs that sequence to get it through the ER. So it will have some type of sequence, but it will probably have like an additional sequence to get it through like the Golgi and everything else in the endomembrane system, which we'll talk about in a second, to get outside of the cell. Otherwise it will stay in the cytosol. So once they leave the ER, proteins rarely come back out again. And this is just important to remember that the signal sequences are the portion of the protein that tell it where to go. So basically it needs that little zip code to tell it where it needs to end up. So here's just a list of some of the signal sequences that I know Laura talks about sometimes in like her lectures. So basically the main point to remember is that signal sequences don't always have to be this exact pattern. So it doesn't have to be leucine, 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 valine. As the pattern you like see here is that these green amino acids are all nonpolar amino acids. So it doesn't have to be exactly leucine, 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 but it can be any combination of nonpolar amino acids in this little stretch right here. And that's pretty much the same for most of them. It's not like an exact sequence. It's just usually the type of protein or type of amino acid that is in that portion of the sequence. So for the ER, you're going to have this long portion of the of nonpolar amino acids, which usually help the cell signal that this is going to the ER. So here's your N terminus because it's found near the N terminus when it first comes out. Here, retention in the lumen of the ER is near the C terminus, but you have a lysine and then two acidic. So basically, the main takeaway here is that each signal sequence is a certain type of amino acid. And this chart is really helpful, I think, to go back to just to give you an idea of the different types of signal sequences. So you have the important to the ER, retention, important to the mitochondria, important to the nucleus, and important to the peroxisomes. There's more signal sequences. This isn't exhaustive, but it just shows how these signal sequences are important for telling the protein where to go. And notice how there isn't one for cytosol. That's because if the protein's going to stay in the cytosol, it does not have a signal sequence. So this chart is super useful. You don't have to memorize it, but it's just very, very useful. A lot of students last semester really got a lot out of this chart. So it shows basically just the life of a protein. So this is the translation, as you know, mRNA then translates the protein. So then you have, is there an ER signal sequence? Yes or no. If there is no ER signal sequences, then you end up in the cytoplasm if there's no other signal. And it's translated by a free ribosome, not a ribosome that's docked to the ER, which would be if it does have an ER signal sequence. And then for the protein, if there's another signal that indicates it needs to go to the nucleus, mitochondria, or chloroplast in plants, it will go there. If not, it'll stay in the cytoplasm. If it does have an ER signal sequence, the ribosome that's translating the protein will dock to the ER. The protein will enter the ER during translation. It'll go straight into it. And then this is an important note. If the protein needs to stay in the ER, it will have another sequence indicating that it will stay in the ER. If not, it'll go into the ER lumen. This one doesn't really show where the ER, where it would be retained in the ER, but there is a signal sequence that indicates that it will be. And then this just goes through all the other endomembrane system organelles, and then how it can be secreted, how the protein would be secreted. So if the protein needs to be secreted, it needs to go through this entire process to be secreted outside of the cell. Same with if it needs to be in the Golgi, the ER, or the lumen. That was kind of a lot. Nikki, do you have any questions?
No, I'm good. Thank you.
Okay. So yeah, here's another version of the flow chart. Basically, it's just helpful to see where it would end up. Just remember that this whole process, so if you need to go through the Golgi, you need to go to if you need to end up in the Golgi, you need to go through the ER. If you need to go to the lysosome, you also need to go to the ER. So this is just kind of helpful for you to identify what is in the endomembrane system. These organelles are not in the endomembrane system. So basically, the main takeaway that this chart shows is that if you're going through the endomembrane system, it's the transport of the proteins occurs simultaneously with translation. That's why the ribosome docks to the ER if the signal sequence indicates that you're going to the ER. But if you're going anywhere else that's not in the endomembrane system, like the nucleus or the mitochondria, transport occurs after translation because the ribosome is not docked to anything. So these two charts definitely recommend going through. You have to memorize them, just to help you identify what the signal sequences indicate. So there can be multiple signal sequences on a protein. So what happens if there is? These are questions, right, Sebastian?
Yeah, these are questions.
Okay. All right. So yeah, Nikki, this kind of is on you. But you can either put in the chat or answer, it's totally fine. But if so, a protein that has an ER localization sequence and an nuclear localization oh my God, I can't speak. Sequence, where will it be located shortly after translation? So these are the type of questions that Laura likes to ask, just a heads up on her exams.
Would it go through the whole endomembrane system because the ER localization sequence is located at the N terminus?
I believe so. I forget.
Yeah, that's actually exactly right.
Yeah.
The ER localization sequence is, as Rachel showed in that chart in the beginning, it's closest to the N terminus. And like you just said, Nikki, the N terminus is the first part to be translated. So the ER localization sequence is the first sequence to be translated. So that's immediately where it's going to go. It's not really going to recognize the NLS sequence.
Yeah. Yep, that's right. Okay. So number 2, a protein has an NLS and an ER retention sequence. Where will it end up?
I'm not sure where the retention sequence would be, but if it doesn't have an ER localization sequence, can it not go through the endomembrane system?
That's also right. Yeah.
Yeah. Good job. Yeah. So yeah, so this is kind of a trick question a lot of kids fall into. They think that it'll be retained, but you need the localization sequence if you're ever going to enter the ER and anyways. So yeah, so it'd end up in the nucleus. Good job. Number 3, a protein has an ER localization sequence, a mitochondrial localization sequence, and a Golgi retention sequence. Where will it end up?
The Golgi?
I think so.
That's right. Yeah.
Yeah. Yep.
Just, I'm like,
Yeah, I was thinking Golgi. I just want to double check. Yeah. Because you have this ER localization, so it'll go through the endomembrane. The mitochondrial localization sequence is, hold on. Why is it not the mitochondria again, Sebastian? It's because it's after translation, right?
Right. It comes after the ER localization sequence. So the ER
Right.
localization sequence is the first localization sequence. If it doesn't have an ER localization sequence, it might have a mitochondrial localization sequence or an NLS or something. So because ER is like the first one, it's the closest to the N terminus. That's immediately where it's going to go, despite it having other localization sequences. And because
Right.
it has the Golgi retention sequence, it'll be retained in the Golgi.
Right, because it's co-translational transport, and that one's post. Okay, yeah. So a protein has an NLS and an NES. What happens?
It would be a protein that like could go in and out of the nucleus. Or like if it's one like I think she was saying like for cell division, like they have the sauce like break down the nuclear envelope, so it needs to keep like that nuclear localization sequence.
Yeah, that's right. That's exactly right. Good job. Do you have any questions on any of these?
I don't think so. Thank you.
Yeah, no
Yeah,
problem.
you
Got them all right.
You're like
So
the first person to ace all of these.
I
like,
know. All right. So this is just a recap of nuclear import and export. Use the names. That's very important because you have these GEFs and gaps that are easy to mix up. So GEFs are guanine nucleotide exchange factors. The role of GEF is to exchange GDP for GTP. And it does this by binding to RAN, which causes a conformational change. So the change in shape reduces RAN's affinity. So remember we talked about affinity last week for GDP, and GTP diffuses into the binding pocket. This is important to be active. This makes the protein active and kind of what is considered to be on. And this occurs in the nucleus. So GEFs are always in the nucleus. That's one thing to take away from this slide if you don't take anything else away. And gaps are always in the cytoplasm of the cell. So they are not in the nucleus. So gaps are basically kind of the opposite of GEFs. They hydrolyze GTP to GDP. So they hydrolyze GEFs exchange. So you can't, it's not adding a phosphate to GDP, it's just exchanging it for an entirely different molecule, which would be GTP. Whereas GAP hydrolyzes GTP to GDP by losing a phosphate. It does this by stimulating the intrinsic GTPase activity of RAN. RAN, this protein I talked about, is a GTPase. So it has this ability to do it on its own, but without GAP, it does it very, very slowly. It does it so slow that the cell like wouldn't be able to function if RAN was really the only thing doing this activity of hydrolyzing GTP. So that's why it needs GAP to help it speed that up. And this occurs in the cytoplasm. So it's turning this GTP, it's turning this like complex off by hydrolyzing GTP to GDP. So the main takeaways here are, if you literally don't remember anything else in the slide, just remember that GEFs are always in the nucleus and GAPs are in the cytoplasm. And we have some diagrams that will show that also. So nuclear import. Oh, you put credit to me. That's so nice. So nuclear import starts with, here, let me put my pointer on. Number 1, so the protein with a nuclear localization sequence binds to important, which is this little circle here. So here's the cargo protein with the little NLS it binds to important. Number 1 occurs right here. So the complex enters the nucleus through a pore, which is this complex right here. And then once it's in the cell or in the nucleus, RAN GDP is now RAN GTP. So this, see, GEF is right here in the nucleus. So while this protein complex is being transferred into the nucleus, GEF is exchanging GDP for GTP. So GTP comes in, RAN's now active. So what happens is that RAN GTP, once it's now bound, after thanks to GEF helping it out, it binds to important, this one, changes its conformation so that the target protein that's needing to get into the nucleus can now be released, and it's on its own, can go off and do whatever its job is inside of the nucleus. And so now RAN GTP is now bound to important because important needs to be bound to RAN GTP to exit the nucleus. So it can continue the cycle of bringing proteins into the cell. So it exits the nucleus once again through the pore. And then for number 5, with the assistance of GAP, which is in the cytoplasm, like we talked about, GAP helps hydrolyze GTP to GDP to release important, so that it can go and bind to another protein with this NLS sequence and take it into the cell for this cycle to be completed. So the main takeaways here, you have your GAP in the cytoplasm and GEF in the nucleus. And you need this RAN complex to take important out of the cell and then release it so that it can continue the cycle. Do you have any questions on this nuclear import?
I did have a question when I was studying. I don't know like if it like matters, but when we say that like GEF only exists in the nucleus, like is that the GEF that binds RAN? Because like I vaguely remember, and I don't know if I'm like mixing things up, but that like a GPCR uses a GEF or like the G protein.
Oh yeah. I know what you're talking about. I don't. Is it RAN GEF in the G, or is that RAS? That might be.
Maybe I might be like mixing things up.
I know RAS and RAN are easy to mix up. I'm not entirely sure.
That's okay.
We'll talk about GPCRs, I think, pretty soon.
Okay.
But just to clear that up, but for now, I would just try and remember that RAN GEF is in the nucleus.
Okay.
I don't think it's what you're talking about. I think that's something different.
Okay, thank you.
Yeah, no problem. All right. And now nuclear export, which is the reverse, basically. So now we're starting in the nucleus with a protein that needs to leave. The protein will have a nuclear export sequence right here, as you can see on this little green protein. So basically what happens is this green protein needs to bind to exportin, which is this little brown molecule, as well as RAN GTP to help it leave the nucleus. So so basically the same process happens. RAN GEF turns RAN GDP into RAN GTP, which allows it to bind to exportin. Exportin can then bind to the cargo protein. So now the main difference here is that you have this complex of 3 proteins together to help it leave through these pores of the nucleus. And then once we're in the cytosol, GAP does the same job of hydrolyzing GTP to GDP. Here's that little phosphate that you lose. And when it does this, it changes the conformation so that exportin and the cargo protein are both released. So the cargo protein can then go off into the cytoplasm like it normally does, do its thing, whatever. And exportin can re-enter the nucleus to bind to another cargo protein. So sometimes people get mixed up, but just to recap, exportin can enter the cell without RAN GTP, and it needs it to exit. Actually, they both can. Yeah, they both can. But yeah, the main difference here is that you have this complex of 3 to exit, because then your cargo protein is released into the cytoplasm. So it's basically the opposite, but RAN, GEF, and GAP are still doing the same things. You're just ending up in a different location. Do you have any questions on this one?
Yeah, I just realized, so in nuclear import,
Mhm.
never like a complex of 3. I just
No.
realized that. I just wanna confirm.
Yeah.
Okay.
So in import, you just have this complex of important and the cargo protein, because then the protein's released into the nucleus. So then important is just the only one that's bound to RAN GDP when it leaves. Whereas export, you have this, because the cargo protein's starting in the nucleus, so it binds to this complex to get out. So now some more questions. Okay, true or false? GEFs are
Okay,
located.
thank you.
Yeah, no problem. GEFs are located in the nucleus during nuclear import and export. Is that true or false?
True.
Yep. GEFs are in the nucleus. GAPs are in the cytoplasm. GEFs phosphorylate a GDP to make GTP. Is that true or false?
No, they bind to the protein and changes conformation so that it no longer has a high affinity for GDP. So then it dissociates out of the binding pocket, so GTP can diffuse in.
Yeah. Yep. Exactly. Good job. Yeah. So yeah, that's important. GAPs release of phosphate, so you can see it better here. GAP releases a phosphate, so it's the same GTP that's turning into a GDP, whereas GEF, you're diffusing in a whole different molecule, as you can see in this little arrow right here. So yeah, good job. So that 2 is false. 3, GAPs stimulate a RAN to hydrolyze GTP to GDP in the cytosol. True or false?
True. Like it stimulates, I think she said like the intrinsic ability of RAN.
Yep, exactly. Good job. And then number 4, GDP is in higher concentration than GTP in the nucleus.
False, and that's why like GDP can diffuse into the binding pocket.
Right, exactly. So yeah, so we have what? True. Number 1 is true, 2 is false, number 3 is true, number 4 is false. Good job. All right, so number 5, what would happen if the KD between important and the cargo protein was greatly decreased?
So a decrease in KD means an increased affinity. So so maybe it would, like just in general, it would pick up more, like a higher concentration of important receptors.
Yeah. So if the KD between important and the cargo protein was greatly decreased, you're right. So the affinity would increase. So it would actually be harder for the important to let go of the cargo protein when it's in the nucleus, because it doesn't want to, now that strong bond between them. So it would just be difficult for the cargo protein to be released into the nucleus to do its job. So you'd probably have like a deficiency of something, whatever the protein's job is in the nucleus.
Okay, that makes sense. Thank you.
Yeah, no problem. And then 6 is the opposite. What would happen if the KD between exportin and the cargo protein was greatly increased?
So the KD was decreased, affinity would increase. So yeah, it would be the opposite. So sorry,
Yes.
I'm just looking at
No,
the
you're totally fine. Yeah. So if the KD is greatly increased, the affinity will decrease between export and cargo, because of that inverse relationship. So it'd be more likely to let go of the cargo protein.
Right.
When it's in the cytosol. I actually don't know if it would let go of it too early. I don't think it would, but I actually don't think you need to know that. I was just thinking about it, but yeah, so the affinity would be lower between the 2.
Okay.
And then number 7, what would be the result of a loss of function mutation to RAN-GEF? So RAN-GEF doesn't do its job.
So GEF wouldn't be able to like cause that conformational shift for to like make the binding pocket available for GTP. So then the cargo wouldn't be able to be released, I guess.
Yeah. So if GEF doesn't do its job, it won't be able to bind to. So you're right. It stay in this off ran GDP state.
Yeah.
So it wouldn't be able to bind to export, and in this case of export, it wouldn't be able to bind to these two proteins and take them out of the cell. And then it would be the same in import. It wouldn't be able to bind to important to take it out of the cell. So you'd probably have a deficiency of cargo proteins entering the cell because they need this important to enter. And then it's like the same with export too. Yeah, good job. All right. A few more. So true or false, a protein can enter in the nucleus while fully folded.
Yes, because like the nuclear pore complex, like they're wider.
I believe so. I just want to double check. Yep. That's true. The nuclear pore is big enough for a fully folded protein to enter. Number 2, a misfolded protein with an LLS may not be able to go into the nucleus.
Ooh, that's interesting. I think so because like the nuclear localization sequence, like it's like a portion of the folded protein. So if you have like all the if you have the atoms or whatever in the protein like shifted, I don't know.
Let me check the answer for that one. So yeah, so this one's true because because the
that makes
misfolded,
sense.
it won't be able to bind to important, right? And then number 3, a protein with an NLS can move back and forth between nucleus and cytosol.
I think it needs the exit sequence or signal.
Yep. Exactly. Yeah, you need that NES also if you need to go back and forth. Just double check. Yep. Okay, yeah. And then number 4, a protein that enters the ER won't go back into the cytosol.
I think so, because I don't remember like a sequence that like would make go backwards.
Yeah. And yeah, once you're in the ER, you usually don't leave unless you're going through the endomembrane system or outside of the cell.
Okay.
Yep. Okay. Good job. Do you have any questions?
Not right now. Thank you.
Okay. Sebastian, your turn.
Okay. What are we talking about? Gene expression regulation. Okay, I guess like the biggest thing to keep in mind with this section is that eukaryotes function very differently from prokaryotes. So kinds of questions, you're fine. Questions that you'll see in this section will often ask you to, I guess, compare the differences between eukaryotes and prokaryotes. So just try to keep some of the differences in mind. I think maybe if not on this slideshow, we have another one that lists the differences pretty well. So if you ever need a reference like a quick difference between the two, you can always check those out. But essentially, I'll be like flipping back and forth, or I'll be comparing the differences between eukaryote and prokaryote gene expression and how they regulate their gene expression. But anyway, so central dogma, I'm sure you're already familiar with this. You have your DNA. If the DNA or if the cell needs some kind of protein, it will make a messenger RNA that codes for that particular protein, and then that gets translated into the protein that was needed. So real quickly, in eukaryotes, transcription happens in the nucleus and translation in the cytosol. There's never any translation in inside the nucleus for eukaryotes, but for prokaryotes, like I was saying, I'll be like comparing and contrasting the differences between the two pretty often. So this is one of the most basic ones, I guess. For prokaryotes, transcription and translation are all done in the cytosol. Bacteria or prokaryotes don't have a nuclear envelope, so it must be done in the cytosol, and it happens at the same time. So that affects different things like mRNA processing, which again I'll talk about later. But that's essentially the central dogma. Do you have any questions about this so far?
Not so far, thank you.
Okay, no worries. You could go ahead, Rachel.
Oh, sorry.
No, you're fine. Okay, so real quickly, which one of these do you think involve gene expression regulation? So here's like a bunch of different steps. Is there any one here that you think may not involve gene expression regulation?
I think all of them, because I think she said like we consider like regulating like protein activity and like degradation also as part of gene expression.
Exactly, yes. So all of these are involved with gene expression regulation. Gene expression, or the regulation of that, isn't just limited to central dogma. So it's not just limited to what genes get expressed, what genes like ultimately get translated. It even goes, like you said, it continues into the life of the protein. So how long does that protein exist? How does that protein get degraded? Where does that protein go? All of those different factors do involve regulation of gene expression. So yeah, actually all of these are involved with gene expression regulation. Good job. Could you go to the next slide, please? I don't think I have control of this.
No, you
If
don't. I
want,
thought for some reason I thought you were gonna go through.
no, it's fine.
No, you're good.
If
You're
you
good.
want, okay. Okay. So the 5 different things. There are 5 different ways that eukaryotes control their gene expression. We won't be talking, like at least in this course, we won't be. I'll mention all of them, but we won't talk about all of them in like great detail. We'll only talk about maybe like 2 or 3 that you should be aware of. But I'll go through all of them just in case, or at least the ones that Laura talked about in her video. So the first way it regulates is chromatin remodeling. Okay, and as you know, the DNA contains the information to code for all kinds of proteins, for every protein. But obviously, your cell doesn't want to express the entire DNA when it needs one protein. So when it does need a specific protein, like let's say it needs I don't know, like some channel protein somewhere, it's obviously not going to transcribe the entire DNA. It's only going to transcribe the part of the DNA that's responsible for that channel protein that it needs. So one way to regulate this is to open that condensed DNA. So you can see here at the top of that picture, there's the DNA in like its closed state. And again, if the cell wants a channel protein, it'll go to where that channel protein is located on the DNA, where the gene for that channel protein is located, and it'll decondense the protein. It'll decondense it so that transcription factors and the RNA polymerase could bind to that site and make the mRNA for that protein that it needs. So again, one of the first levels of regulation is just condensing or uncondensing or loosening the part of the DNA that it needs opened. So true or false, prokaryotes and prokaryotes have very similar transcriptional control. What do you think?
I think because prokaryotes don't have a nucleus, they don't organize their DNA and chromatin.
Right, so this statement's false. Their transcriptional control is very different. They have circular DNA or they have a chromatid. So they still do some level of transcriptional control, but it's very different from how eukaryotes control their transcription. It's very different from this condensing and decondensing. So they still have some level of transcriptional control, but definitely not to the same extent that eukaryotes do. So yeah, that statement is false. Okay, you could go ahead, Rachel. Sweet. Okay, so I just mentioned how DNA could either be condensed or decondensed. You want to decondense your DNA if you need a particular gene translated. Another way that you could control this is by transcription factors. So you have your RNA polymerase, which of course is what does the transcribing. It's what makes the mRNA transcript. But before the RNA polymerase can actually bind to the DNA, it needs transcription factors to help that. So the RNA polymerase cannot bind without the help of transcription factors. And you have several different kinds of transcription factors. Some of them are responsible for promoting transcription. So some of them help the DNA polymerase transcribe, and other of them like suppress it. So you have an array of different transcription factors. Again, for the purpose of this course, we're not going to be talking about like every single one extremely meticulously, but just so you know that you do have transcription factors that control this rate of transcription. So each of these are obviously pretty important. Okay, so any questions about this so far?
I'm good, thank you.
Okay. Okay, so another thing that cells could do is they have the RNA modification. So again, in prokaryotes, transcription and translation happen at the same time. So RNA modification doesn't exist in prokaryotes. This is another one of those differences. It doesn't exist because, again, transcription and translation happen at the same time. So you don't really need to modify your RNA. But in eukaryotes, obviously, we have mRNA. The mRNA, like we just mentioned, it has to be transcribed in the nucleus, but that mRNA needs to find its way into the cytosol, which is a much harsher environment. So you do need to modify it. You do need to keep it stable. It needs to travel in the cytosol to find that free ribosome, all that stuff. So here's how eukaryotes modify their RNA. So after the transcript is made, the first step that they do, I don't know if these steps are in order. They're probably not. I think it could happen in any order, but these are essentially the three things that need to happen. The guanine cap needs to be covalently attached. The poly A tail needs to be attached at the, the guanine cap is attached at the 5' end. It's covalently attached. The poly A tail is attached at the 3' end. And the attachment of these caps at the 5' and 3' end is just essentially to increase the stability. So once, obviously the mRNA needs to travel from the nucleus into the cytosol. One way to keep that transcript or that RNA stable is to add these two caps. So these caps are essentially just responsible for adding stability to your transcript. And then finally, you need to splice out the introns. Introns are these stretches of non-coding nucleotides. So of course you need to get those out before you could have a mature mRNA. Okay, so any questions about that so far? No, you're fine.
No.
Okay, if not, I have a question for you. So let's say you're in this lab and you isolated your mRNA and you accidentally boiled it somehow. You took it out of that boiling pot and then you had it translated in some free ribosome. However, now you're noticing that for whatever reason, after you boiled it accidentally, your protein is not coming out correctly. There's something wrong with it. And you're thinking that the boiling may have messed up the 5' cap. So what do you think about this? What do you think about this speculation? Do you think that's a good argument to make, that maybe the boiling messed up the 5' cap, or do you think maybe the problem could be elsewhere?
I don't remember like if I'm getting confused between the poly A tail, but I don't know like if translation would still occur without the 5' cap, or like if it would occur, but maybe now because you don't have that 5' cap, like you can have endonucleases like chewing off like bits at that end, and so now you're missing like some of the amino acid sequence.
That's a good argument. That's a good argument, actually. You don't think about that. Yeah.
But
Okay.
also, I don't remember like I know like I don't think, or maybe I'm thinking of the pre-mRNA where like that like some of that doesn't get included in the final thing because like the promoter is like located farther down, but I don't remember.
Yeah. Yeah, that's actually a pretty good argument. This student is suspecting that the boiling damaged the 5' cap, and that's ultimately what's causing the problem. However, this isn't the best argument to make, only because the 5' is covalently attached to the RNA. So boiling is not really going to mess up. Boiling could mess up your hydrogen bonds, which are considerably weaker than your covalent bonds, but boiling isn't, you're going to need a lot more things to break a covalent bond. That's a very strong bond that boiling generally doesn't mess up. However, what you said, that could be a good argument. If somehow the 5' cap was removed somehow, and endonucleases did start chewing up your RNA transcript, then yeah, you could totally lead to a messed up protein. But right now, if you just consider like these three factors, I'd say the main thing leading to the problem would be the intron splicing. Splicing out your introns seems like that would be the cause of the problem, only because the boiling doesn't really have an effect on the 5' cap. Like you can't remove a covalent bond with just boiling. You're going to need either like an enzyme for that, or you're going to need like some extremely high heat that surpasses boiling by an extreme amount. Same thing goes for the poly A tail. It doesn't really seem like that might be a problem. So in this scenario specifically, it may seem like it might be the intron splicing that may be the problem. if you're only considering like these three things. But of course, there, like you just mentioned, there could be several possibilities for what went wrong with this.
Thank you. That makes sense. I think I missed that the guanine cap was covalently attached. So that makes a
you're
lot
fine.
of sense.
Sure. Okay. Okay. Another way to control your gene expression is by altering the life of your mRNA. So mRNA has a half-life. It can only exist for some amount of time. It's different for every mRNA transcript, but it can only exist for some amount of time. If your cell wants to get rid of mRNA, it would probably have like a relatively low half-life, so it could get rid of it like pretty easily. So for instance, if it's a protein that you need for only a short amount of time, it would probably have like a very low half-life just so it could get degraded pretty easily. But if it's a protein that you need around carrying out a lot of functions for a really long time, it might seem more likely that the mRNA transcripts has a much longer half-life. Again, this is one of those that we're not really going to talk about too much in this class, but just so you know, this is one way that a cell could regulate its gene expression. Another way is mRNA localization. So where the mRNA is transported in the cytosol. Most mRNA transcripts go to the free ribosome so that it can be translated and it'll go through all those steps that we talked about in the previous part. But let's say a cell transcribed way too many mRNA for a protein that it actually didn't end up needing all of those mRNA transcripts. One thing that it could do is, instead of sending it to a ribosome to be translated, it could send it to a P-body for it to get degraded. This particular step, like the mRNA localization to a P-body or something, we're not going to talk about so much now. That this actually becomes more relevant when we talk about RNA interference. So as of right now, this isn't too important, but we might come back to this when we start talking about RNAi. Any questions about these, by the way?
I just forget what a P-body is.
So did I.
Yeah, I don't know.
It's like this, it's kind of like this bubble where nucleic acid, or like mRNA or any like nucleotides, any ribonucleic material, it goes in there and it essentially gets recycled for it to use something else. So in this example, if the cell transcribed too many mRNA that it actually didn't need, it would send it to a P-body so that those nucleotides don't go to waste. It'll deconstruct the mRNA transcript. It'll break everything apart, and then the cell could just recycle those nucleotides and use it for something else, essentially.
Thank you.
Yeah, no problem. What is this? Oh, okay. One of the last steps of gene expression control is whether or not it gets like translated. So I think this. Oh, okay, yeah. So this ties back a little bit with like the fourth step. So once mRNA leaves the nucleus, it could be either sequestered to a free ribosome for it to get translated, or it could be sequestered into the P-body. There actually is a regulatory factor that goes into the rate that mRNA gets transcribed. So let's say the cell really needs this mRNA to get transcribed as soon as, translated as soon as possible. It would essentially have a very high rate of translation for that mRNA. But if it's a protein that the cell doesn't really need, but it might need like maybe later on, so some protein that isn't extremely crucial at the time, it might have a much lower affinity to the ribosome, or it might have a lower translation rate. So translation rates essentially are another level of regulation that the cell could go to. Okay, any questions about that so far?
I think I'm good, thank you.
Sweet. Okay, so this is eukaryotic gene expression. I remember this question specifically when we had in-person exams. Laura or Professor Francis gave this exact diagram, except she took out all of the words in this diagram, and she asked you to label the steps onto it.
I think that was on the practice questions because
Oh,
I
was
vaguely
it?
remember that. Yeah.
Yeah. Okay. Yeah, Rachel, you might remember this one.
Oh yeah, no, I remember that.
Yeah, I was like,
I remember that.
Yeah. So in eukaryotes, again, this is slightly different from prokaryote gene expression, which I'll talk about in the next slide. But okay, this was in your problem set, I think you mentioned, so I won't go through this extensively, but this is like a good little diagram about the life of mRNA, its lifespan, I guess. So it goes from DNA to the mRNA transcript, it becomes an mRNA sequestered into the cytosol, and a bunch of different things could happen to that mRNA transcript. It could get immediately degraded, it'll most likely become a protein, and then that protein has its own life. It has its own level of regulation. Yeah, so again, I know you just studied this in your problem set, so I won't go through it like extensively, but if you have any questions about this diagram, feel free to let me know, of course.
Thank you.
Sure. Okay, this is prokaryote gene expression, so it's slightly different. Again, for prokaryotes, they have very different, they still have transcriptional control, but it's very different from the way the eukaryotes transcribe it. And I think I mentioned before, there literally is no RNA processing. In RNA processing is just like the creation of mRNA. So adding the 5' cap, adding the poly A tail, taking out the introns. Prokaryotes transcribe and translate at the same time. So they don't really need to process anything. So this step 2, RNA processing control, that doesn't even exist in prokaryotes. Same thing with step 3. Step 3 doesn't exist because, again, prokaryotes don't make mRNA. So they have nothing to transport. They have nothing to localize to a free ribosome or anything like that. They don't leave the nucleus because, again, prokaryotes don't have a nucleus. So everything past that, everything past like this mRNA step. So yeah, degradation of mRNA, I probably should have taken that out. But like the protein being degraded, protein being activated or inactivated, prokaryotes do have all those steps though. Okay, so here's some practice problems. So what level of gene expression regulation does not affect prokaryotes? What do you think?
I think she said, let's see, like, was it protein degradation, like, evolved to, oh no, that was the endonucleases evolved to protect against RNA viruses. So yeah, I forget if protein degradation is in prokaryotes, but B would, mRNA, like you just said, no, and protein deactivation would.
Yeah, so all of these except for B affects prokaryotes. Prokaryotes, obviously, they of course make their own proteins. And like you mentioned before, gene expression regulation goes all the way into the life of the protein. So in prokaryotes, protein degradation is a level of control. So it does affect prokaryotes. Prokaryotes don't have mRNA, so that essentially won't affect it. Wait, I'm sorry. What level of gene expression regulation does not affect prokaryotes? Yeah, it would be either B or C or both, because the protein's life is still affected in prokaryotes, but translation and protein synthesis, again, it happens. Gosh, what am I trying to say? Because these things happen simultaneously, it's not a tremendous factor for prokaryotes. Sorry, I like just
That
lost
makes sense. No, that makes sense because it's coupled with transcription. Like,
right. So in one of the steps before, at least for eukaryotes, I mentioned how the rate of translation is one of the transcriptional controls for eukaryotes. But again, because transcription and translation are happening at the same time for prokaryotes, translation rate or protein synthesis, that rate doesn't really affect prokaryotes. Neither does mRNA degradation, because again, prokaryotes don't make mRNA. Okay, Does that answer any questions?
Yeah, that makes sense. Thank you.
Sure, no worries. Okay, and a mutation occurs where the poly A tail is not attached. Which of these do you think might happen?
A?
Yeah, that's right. Sweet.
That's it.
Nice.
That's all we
Thank you,
have.
guys.
Yeah, of course.
Anytime.
Okay. Yeah, if at any point you have like any questions about something, or you know, feel free to message any one of us. We'd be happy to answer those.
Thank you so much. Have a good night.
Yeah,
Thanks,
you
you
too.
too.